Hello students, in today's session we'll discuss DNA fingerprinting. DNA fingerprinting. You already know that DNA fingerprinting, which is commonly used to identify the unique difference among the individuals and also used to identify the paternity issues. There are so many applications, and in the last session we discussed about a human genome project. And you already know that 99.9% of human DNA, which does not code for any proteins, and only 0.1% of human DNA codes for proteins, and 99.9% of base sequence among the human beings is same. These are the non-coding sequence. It is same in all the individuals in a population. Then the question arises: the differences in the sequences which makes an individual unique in their phenotypic appearance. So 99.9% of base sequence is same in all the human beings, and only 1.1% of base sequences which makes the individual unique in their phenotypic appearance. For example, when you compare parents and offsprings, you find the differences, and differences occur in terms of color, shape, size, etc. When you compare among your relatives, you find some differences. So those differences are unique to the particular person. These differences, to find out these differences among the individuals or between the two individuals in a population, it's very difficult to identify the differences. For example, if you go for DNA sequencing every time, it's a very expensive task, because you already know that in Human Genome Project, we already discussed about in the Human Genome Project, so that the human genome is made up of 3.3 10 to the power 9 base pairs. The cost for one base pair is US $3. So if you want to identify the differences through DNA sequencing, it will cost about US $9 billion. $9 billion. So it's a very easy technique to identify differences, such differences in an individual or within a population, is through DNA fingerprinting technique. So through DNA fingerprinting is a very quick way to compare the DNA sequences of two individuals. And DNA fingerprinting, which helps in identification of individual at a genetic level. I told you, you find the differences between parents and offsprings, or you find variations among the parents, and when compare the parents and offsprings, you find variations at the genetic level, and these variations occur in some regions of DNA sequence, not in all the regions of the DNA, some regions of DNA sequence. Such DNA sequences are called repetitive DNA. You got my point? Okay. For example, I'll write it here. If, for example, the parental repetitive sequence A, G, C, T. A, G, C, T. This is case 1. In offsprings, you find A, G, T, T. So look at here. So when you compare the third nucleotide base in parent, okay, in father you find AGCT, whereas in case of child or son or daughter, AGTT. In this region you find the differences, and these differences are unique to the individuals, and these sequences are repeated n number of times. Okay, for example, 100 to 1000 times these sequences are, okay, repeated so many times. Such DNA we called as a repetitive DNA. Repetitive DNA. So this repetitive DNA is a basis for DNA fingerprinting, or the repetitive DNA is a basis for genetic mapping, one, and it's also basis for DNA fingerprinting. Let us discuss. So repetitive DNA. Come back to the repetitive DNA. So in human genome has 99% of non-coding DNA, and only 1%, 1%, less than 1% is a coding DNA itself. So look at the DNA. Very negligible amount of DNA which codes for protein, and remaining part of the DNA, okay, remaining part of the DNA has a non-coding DNA, and that non-coding DNA has a DNA sequences, and those DNA sequences are repeated so many times, and such a DNA we call it as a repetitive DNA or repetitive sequences. And these repetitive sequences, they are commonly called sat DNA or satellite DNA. So DNA, repetitive DNA, is also known as sat DNA or satellite DNA. So satellite DNA is one of the component of centromere and telomere of the chromosome. Centromere and telomere of the chromosome. Is that clear? So look at here. So it is mainly concentrated in centromere and telomere of the chromosome. Next, the repetitive sequences in satellite DNA, they are commonly called tandem repeats and or variable number tandem repeats. For example, I'll take DNA from human beings, one species 1, I'll take the DNA, and I'll take the DNA of second species, a horse, a donkey, etc. And I'll treat both the DNA with a restriction endonucleases. So the restriction endonucleases which cuts the DNA into fragments. So both the DNA are treated with restriction endonucleases. For example, in case of human beings, that restriction endonucleases cut that particular DNA into fragments. Whereas in case of horse, it cut into 8 fragments. In case of donkey, it cut into 5 fragments. So those repetitive DNA, okay, the length varies. Such DNA we called as a variable number tandem repeats and variable number tandem repeats. Is that clear? So variable number tandem repeats is a base for DNA fingerprinting, and this variable tandem repeats are mainly classified into 2 types based on the base composition, that is purines and pyrimidines, 1, then length of the segment, second one, and third one is repetitive units. So tandem repeats are 2 types. One is microsatellites, 1, The second one is mini satellites. Mini satellites. So difference is based on the number of base space or repetitive units. Okay. Then mini satellites, often called as a variable number tandem repeats. I took an example of horse, donkey and human beings. Variable number tandem repeats. Okay, and these variable number tandem repeats which exhibit polymorphism. Polymorphism. We'll discuss later what is polymorphism. Okay, so the polymorphism, DNA polymorphism, is a base for genetic mapping and DNA fingerprinting. The question arises, how we can separate repetitive DNA or satellite DNA and bulk DNA. So this DNA is separated during density gradient centrifugation. So as a result of during density gradient centrifugation, we could able to get two peaks. We could able to get two peaks. Okay, the major peak we called as a bulk DNA, and the smaller peak we called as a SAT DNA. And this SAT DNA is further classified into two types. One is microsatellites, another one is mini satellites, and these mini satellites are commonly called variable number tandem repeats. Variable number tandem repeats. Next, the mini satellites are unstable and mutable. So you already know that. I told you about when you compare parents and offsprings, you find variations in the characters. So you find a number of variations when you compare parents and offsprings. In some region they are similar. In some region, some sequences are unique to the particular individuals. And these repetitive sequences undergo mutations. And you already know that. So mutations occur in both the cells, somatic cell as well as germ cells. So even though in not only in the coding sequences, in non-coding sequences also mutations takes place. So mutation is not restricted to the coding sequences of DNA. It is also takes place in non-coding sequences of DNA. Non-coding sequences of DNA. So that results in DNA polymorphism. I'll show you one example. Okay. For example, A, B, C, D, E, F, G. This is one gene. Okay. So here the mutations takes place in repetitive DNA. What happens? A, B, C, D, E, F, G. In this portion, okay, the nucleotide is added. Okay, one nucleotide is added. This is called single nucleotide polymorphism. So such mutations which takes place both somatic cells and germ cells. And in case of germ cells, these such mutations does not impairs the ability of individuals to reproduce. But these are inheritable mutations. They are transmitted from one generation to the other generation. The frequency, higher rate of frequency, okay, if the variations occur in such a DNA sequence, that is repetitive DNA, if inheritable variations occur at higher frequency, then it is called DNA polymorphism. Okay, I'll repeat once again. If inheritable mutations observed in a population at higher frequency, higher frequency, it is referred as DNA polymorphism. So next one, so DNA polymorphism, or we commonly called genetic polymorphism. So variations in the DNA sequences, DNA in the genome. So DNA from any tissue of individuals will show same polymorphism. It's the same. In some regions of the DNA you find changes. So polymorphism is heritable. I told you some changes takes place. When you compare this one and this one, some changes will take place that results in DNA polymorphism. DNA polymorphism. So these changes are heritable and mutables. So heritable and mutable, they results out mutations. Such mutations, this is an example, A, B, C, C, D, E, F, G. So one nucleotide is added to that repetitive DNA that results in polymorphism. So such mutations occur, accumulates within the population generation after generation, and does not affect much more because they are not related to the coding sequences, does not affect the production of proteins, but those variations accumulates accumulation within the population and transmit generation after generation, and that results in evolution, or that results in speciation or origin of new species. So look at here. So it may be takes place single nucleotide to multiple changes. One nucleotide change ಆಗಬಹುದು ಅಥವಾ ಬಹಳಷ್ಟು nucleotide changes ಆಗಬಹುದು. Okay, that results in DNA polymorphism. DNA polymorphism. Look at here. CTC is a one repetitive sequence, and CAC. So thymine is replaced by adenine. Thymine is replaced by adenine. So this is we called as a single nucleotide polymorphism. Single nucleotide polymorphism. So polymorphism is important in evolution and speciation. And polymorphism is a base for genetic mapping. Okay, it is genetic mapping and also helps in DNA fingerprinting, identifying genetic diseases, and forensic medicines. Is that clear? So the base DNA polymorphism is a basis for genetic mapping, one, or genome mapping, second one is DNA. Start with the technique which is commonly used to identify the individual at a genetic level, the technique which is commonly called DNA fingerprinting. So this technique was first developed by Alec Jeffrey. It is commonly known as DNA fingerprinting is commonly known as DNA profiling method. So this technique was developed by Alec Jeffrey. So definition: it's a technique to identify an individual at a genetic level. Alec Jeffrey. The steps. Okay. Next we'll discuss the steps which are involved in DNA fingerprinting. The first step which is involved in DNA fingerprinting is collection of biological sample. Very important. So cells are collected. Okay, the first one, cells, biological samples. It may be blood, semen, hair follicle, saliva. They are collected as a source of DNA. Samples are collected. The second very important step is from those cells isolation of DNA or extraction of DNA. The question arises, how we can extract DNA from the cell if cell is treated with various enzymes. Okay, so various enzyme, easily we could extract the DNA from the cells. Look at here. So these are the cells. Okay, cells. They are treated with various enzymes that digest the membranes to release chromosomes. From the chromosome, DNA is isolated. DNA is isolated. Look at here. If the amount of DNA is very less, very negligible, that DNA is multiplied through PCR technique, polymerase chain reaction, which is commonly known as polymerase chain reaction. Through polymerase chain reaction or PCR, okay, the DNA is multiplied. Polymerase chain reaction, DNA is multiplied. So enzymes released. So look at here. So polymerase chain. The third step is fragmentation of DNA. So DNA is isolated and it is multiplied by PCR, polymerase chain reaction. That DNA is fragmented with the use of restriction endonucleases. So DNA sample is collected and it is treated with the restriction endonucleases to obtain different fragments, a length of DNA fragments, different lengths of DNA fragments. Such different lengths of DNA fragments, they are commonly called RFLP, restriction fragment length polymorphism. So this is the third step. Fourth one. So as a result of fragmentation, we could able to get the DNA fragments with various length. Okay, various length. So those DNA fragments are separated along the length, okay, through gel electrophoresis technique. Through gel electrophoresis technique. So what we'll do? Okay, we'll transfer, okay, DNA fragments on agarose gel. Look at here. So the DNA fragments are, okay, transferred to the agarose gel. They are loaded to the agarose gel and will apply electric field to the gel. Okay, so the electric field has two poles, okay, anode and cathode. And you already know that DNA is being negatively charged, it moves towards the positive side. When you apply electric field, okay, they move towards the positive sides. So as a result of this, okay, after applying electric field, the fragments move through agar based on their size and density. So as a result of this, when you apply, the movement of fragments takes place. So the smaller fragments, okay, fragments travels greater distance than the larger fragments, and this technique we call it as a gel electrophoresis. So after gel electrophoresis, what we'll do? Okay, these DNA, double-stranded DNA, are converted into single-stranded DNA. On the agarose gel, double-stranded DNA fragments are converted into single-stranded DNA. The question arises, how we can convert these double-stranded DNA fragments into single-stranded DNA fragments? By simple technique. When, okay, agarose gel media is treated with alkali solution, as a result of this, what happens? Double-stranded DNA fragments are into single-stranded. Look at here, single-stranded. So single strand. So its gels are soaked in alkali to make a DNA single-stranded. Single-stranded DNA is transferred to the nitrocellulose membrane. Such single-stranded DNA fragments are transferred to the nitrocellulose membrane. So what happens? This technique of transferring DNA fragments from agarose gel media to nitrocellulose membrane is called southern blot. Southern blot. Look at here. So those DNA fragments, single-stranded DNA fragments, are transferred to the nitrocellulose membrane or nylon membrane by a technique called southern blotting. Southern blotting. So southern blotting is named after the discoverer Edwin M. Southern. Is that clear? So these DNA fragments are bound to the membrane exactly as in the gel. Gel. So after transferring, again the single stranded DNA fragments, again they are converted into double stranded by using known radioactive probes. Probes are nothing but known DNA fragments or nucleotide sequences. This is tagging of radioactive probes is based on the principle of complementary base pairing. For example, I have in the fragments have sequences like adenine, guanine, cytosine, and thymine. Adenine, guanine, cytosine, thymine, and I have a radioactive probes. So thymine, cytosine, guanine, adenine. If I add these radioactive probes to the nitrocellulose membrane, they immediately go and bind to these DNA fragments. As a result of this, the single-stranded DNA is converted into double-stranded DNA. Double-stranded DNA by using radioactive probes. Look at here. So DNA, nitrocellulose sheet are exposed to the radioactive probes, known DNA fragments. The probes are radioactive single-stranded DNA fragments known as a base sequence, and DNA probes will bind with the complementary fragments on the southern blot or the nitrocellulose membrane. As a result of this, what happens? Again, single-stranded DNA fragments are converted into double-stranded. So binding produces fragment probe hybrids. So this is we called as a fragment probe hybrid synthesis. Is that clear? So after tagging with the radioactive probes, we go for autoradiography. So autoradiography is what? Such DNA fragments are placed on an X-ray film, or X-ray film is placed on a nitrocellulose sheet to obtain autoradiographic print. So those prints are used to identify individual at a genetic level. So look at here. So this is X-ray film developed after radiography. Such DNA processed to observe position of the bands of radioactivity. Then, so soon after autoradiography, the fragments are readily available, identified. Some minor changes, or so those minor changes are readily available, on the X-ray films, and those minor changes, or particular fragments of DNA helps in identification of individual at a genetic level. So after autoradiography, we go for look at here. We go for comparisons. Such patterns of bands in the DNA fingerprinting is unique. Okay, so when you compare parents and offsprings, okay, offsprings, they are almost similar. Okay, in some position, okay, some position of the DNA, the bands are similar. For example, when you consider this, okay, this is a parental, okay, bands. For example, father, this is father's DNA fingerprinting, 1, 2, 3, 4. So 4 bands are available. When look at the son's, okay, fingerprint, so some regions that is similar. For example, in this region, okay, it is more or less, it is similar. Okay, similar. So easily we can identify father and son through DNA fingerprinting. So comparison of DNA fingerprints. So provide information of degree of similarity and differences. So this completes the steps involved in fingerprinting. More and come back to this some important points. Population is thus polymorphic for a restriction fragment pattern, and this restriction fragment fragment is length polymorphism, RFLP we call it as a restriction fragment length polymorphism. So this helps in identifying particular gene, particular individuals. Okay, so restriction sites, the distance between the restriction site of two different individuals are different. They are not similar. They are unique to the particular person. Okay, they are person. Next, come back to the applications. The last part, the applications of DNA fingerprinting. So the first one, which helps in crime detection. So crime detection, to identify the criminals, DNA fingerprinting is done. One, okay, forensic science. And second one, disputes or parental issues, paternity issues arises, parental disputation or disputes arising from babysitter. Those are identified through a technique called DNA fingerprinting. Immigration cases. Okay, so if you store database of one country, we can easily identify the immigrants from the other country. Okay, other country. So immigration cases, to solve immigration problem. Gene mutations and its effects are studied. Then to study bacterial and viral infections, to identify parents of the individuals and detect the these are okay, these are some applications of DNA fingerprinting, and this completes the topic molecular basis of inheritance. Thank you.